Good afternoon. Today we are going to present about the cell-assembling proteins hydrogel with modular integrin binding domains. So first, let's talk about the flowchart of this research article. So we produce a repeating amino acid sequence using scramble codon principles, and then there will be an integrate of 3 tripeptide RGD sequences into the predetermined site within the predict disorder coin of the WPT2 protein design. And to test this approach for assembling the modified repeating units are also required as well. So we divide our presentation into 4 parts, which are the introduction, material methods, result analysis, and conclusions. So first, let's talk about the introduction. What is the hydrogel? The hydrogels are the biomaterial that can retain water and soluble proteins. We can control the design of it by the viscoelastic and its structures. We can also enhance the structural scaffold for the cell culture as well. For the material methods.
For the material method, we can't do the PCR because DNA containing multiple of repeating sequence is difficult and can complete fail or produce a heterogeneous final product in the annealing process. As a result, we use the codon scrambling strategies to take advantage of the degenerate relationship between codons and amino acid. And here are the RGD sequence. There are 9 repeats of AGA, GAG, PEG. And here we added 3 RGD sequences, as you can see in the pictures. The terminal A and B leucine zipper domain of WPT2 genes are retained. Basic and acid leucine zippers form a robust hydrogel stabilized by a favorable electrostatic interaction. And this is how we form the WPT2 3R genes.
There will be a total of 6 topics that we're going to discuss in these presentations. So first, let's talk about the protein synthesis and purification. Next slide, please. For the protein synthesis and purification, there are several components that require, which are the synthetic oligonucleotide, the PUC19 cloning vector, and platinum PFX DNA polymerase. Also, the XR1 gluconing cholesterol and the NTP, which is the substrate. And IPDT, which is inducers, the DNA purification kits, and NTA resins and E. coli strain, and other chemicals too. For the purifications, we need to culture the cells and do the cell lysis. After that, we do the column chromatographies and gel electrophoresis. So there are several types of the column chromatographies. Next slide, please. Just as the ion exchange chromatographies, the site exclusion chromatography, and affinity chromatographies. But in this research article, they use the affinity chromatographies. The affinity chromatography is the method of separating a biomolecules from mixtures based on the highly specific macromolecule binding interaction between biomolecules and other substance. The specific type of binding interaction depends on the biomolecules of interest. Next slide, please. So the leucine zipper. The leucine zipper is the DNA binding protein which normally found in plant, human, and vertebrates. And the C terminus of the DNA binding protein consists of the structure called leucine zipper, which can form the coil of a polypeptide chain that is attributed as a 2-chain alpha helix of homodimer and heterodimer. Each teeth of the zip is the order of the amino acid, and every 7 amino acid will be the position of leucine that is hydrophobic, which will order outside the polypeptide chain. At the end terminus, there will be an leucine zipper will connect to the DNA domain that consists of lysine and arginine, which is going to attach to the operator gene as well.
For the protein synthesis, the WPT2 3R gene is ligated into the PQE9 expression vector and transformed into the E. coli using the heat shock method. Transformed cells are spread onto the agar plate with both ampicillin and kanamycin for 16 hours at 37 degrees Celsius. A positive transform selected and used to inoculate 10 ml of LB medium with ampicillin and kanamycin in shaker. Incubator at 37 degrees Celsius and 300 RPM until the OD reaches of 0.6. Cells in 2 ml culture were harvested to produce a glycerol stock. For the last scale, the glycerol stock is inoculated in the ratio of 1/50 in the LB medium, Batch mode fermentation conducted, and the cells are harvested and lysis for the next step.
Next is the purifications. The purification is achieved by performing the FPLC with nickel NCA resins. Then we filter out the supernatant and load it into the NINCA resins. Column with 5 column volume of buffer B. The column was washed with 6 volume of buffer B and C. The his tag WPT2R was diluted by the continuous stream of buffer E. Then we will perform the gel electrophoresis in order to determine whether the protein of interest or the fraction of protein that we purify is correct or not. For the procedure, the WPT2 are the disordered coin domain flanked by an acidic and basic leucine super domains. They have sequence at the upper part of the slide, and we add the RGD at the end. After that, they also contain the histidine tag and also enzymatic cleavage site at the N terminus and C terminus with the cysteine residue as well. The XL1 blue committing cell will transform to the PUC19 WPT2-3R by the heat shock at 42 degrees Celsius in order to make the pore at the cell membrane. And the transformant will culture on the agar plate under the ampicillin resistant at 36 degrees Celsius for 16 hours.
For the circular dichroism, this is method to determine the protein's secondary structure. It is based on the differential absorption of left and right circular polarized. Next slide, please. Diffusion-based spectroscopy is an advanced light scattering technique used to extract mean square displacement of particles dispersed of a solvent from the measurement of the temporal fluctuation of background scattered or transmitted light for viscoelasticity of the protein hydrogel. Next, please.
The macroscopic viscoelasticity of the WPT2-3R hydrogel were investigated in cone and plate geometries using a stress control rheometers equipped with the pulsar temperature control device. After that, the sample of pre-made hydrogel were loaded to the 25mm cone and plate tubes, and the linear dynamic gestures was obtained from the operating rheometer in a control state and feedback modes. Next slide, please. The cell culture. The cell culture is we obtain the human foreskin fibroblast or HFF from the APCC RPMIs 1640 medias. After that, we incubate it at 37 degrees Celsius and harvest with solution of trypsins. Then we resuspension the cells until we sterilize by the UV illumination and rinse 3 times with the PBS. For the cell viabilities, the cell viability is the step that we evaluate the HFF viability on our protein by the cell tire 96 aqueous one solution cell proliferation assays. For the assay preparations, we took 100 microliter of cell were loaded into the protein coated well at the density of 7,500 cells per well. The plate we then remove from the incubators. And we add the mixture of 100 microliter of RPMI, 20 microliter of tetraammonium salts, and MTS to each of the well. And then the plate was returned to the incubator for a period of 4 hours.
Okay, next is the result and analysis. So let's see the first one. Next slide. First is the protein synthesis. As we know right now, what is the structure of WPT2-3R? How are we gonna know that this protein is purified or not? So we have to check by 2 things. That is a reverse phase HPLC, that to make sure that the purity of protein has at least 96%. And also the second, we have to check that is a MALDITOF to check that the molecular weight of protein was 24,597 Dalton or not. Okay, next slide. Next slide, we will check about the secondary structure. We check that there are any chain of secondary structure between these 2 proteins or not. And we use a technique called CD or circular dichroism that mentioned above. And you see the result from the graph between 2 proteins at 222 nanometer in wavelength. You see that residual molar electricity hasn't changed. So you can conclude that the secondary structure is same. And moreover, both of them are insensitive to pH and temperature.
Okay, So now about the hydrogel viscoelasticity. So we're going to measure the shear modulus and look at the effects of temperature because we want to check whether the gel has desired properties of hydrogel that we created. So we use the DWS microRheology in the methods that my friend mentioned. So let's look at the result of the first graph. The this distinct region is important because you can measure the shear modulus. So if you try to look at it closely, you can see this graph and this graph, this line. This both line is the shear modulus. The G prime is the elastic-like behavior, this one, and this is the liquid-like behavior. And this results is consistent with the shear modulus of the microRheology measurement, which means our hydrogel is good in this viscoelasticity properties. Now this is the relaxation time. You can see this, you vary the temperature here. This is at 60, 37, and 27. If you, I'm sorry, this is 27. If you increase the temperature, the graph shifts here, means you have a lower relaxation time. Okay, let's look at the protein fibroblast interactions. So why do you study this? Because you want to know the potential cell and tissue culture application. You use a spectrophotometer and you measure it. Now let's look at the absorbance. If the absorbance is high, that means that your cell is alive, right? You have this bar is a positive control. So this is the modified one. This is the non-modified one. At 6 hours, you see that the modified is close to the positive control, which means that the surface, the cells grows well compared to the one without our modification. So this is another picture to show the growth. You can see this is a glass coated with our modified protein. This one is a surface with the modified protein. This is without. As you can see, there is a obvious cell growth because the surface is well spread and you have a polarized morphology compared to this rounded shape, which lacks the biochemical signal and also inhibit the absorption of extracellular matrix. So yeah, it promotes fibroblast interaction, our modified protein.
Okay, for the conclusion, after that we know that what the protein WPT23R is, what is the advantage of this protein is. So for the first one, this protein supports the adhesion, spreading, and polarization of human fibroblast cells on coated surfaces. Second, there are no effect to secondary structure, mechanic property of hydrogel, and report elasticity of hydrogel. And lastly, it's easily to tailor to use in many applications like biomedical applications. And so that is all for our presentation. Thank you.